All right, this is where we ended up last week, right here. This is where we wrapped up. And I asked you to explain why the spinal cord is considered part of the central nervous system. And the answer really is in what type of information does it process? It makes the decisions for which type of responses. Which type of responses, folks? Reflexes. That is the best example you can provide. It receives sensory information and makes a decision of a motor response. Reflexes are the primary information that it completely processes, a whole circuit without including the brain. What type of neuron runs through this part A? Afferent or efferent? Afferent and afferent only. That is the dorsal root. It does afferent only. Sensory information only. What about B? That is the ventral ramus. What does that one do? What does that one do? I think I heard it. Both. That one is afferent and efferent. We have to understand how the spinal cord is set up and arranged. The ventral ramus is both afferent and efferent. The nerve plexus, we're going to go over them in the end of today's lecture. But before we do that, I just want you to be aware that each of these that comes out in purple is a spinal nerve root. So these are often called roots. Then look how complex it is. These interparts, I don't need you to know. The roots come together to become trunks. Then they split to become various branches. Then they come back together to become, I almost forget what these are called, rami. So they like join together, they split, they join together again before they become what we know as the peripheral nerves. The nerves that go to the body. Remember that at the roots, they are already afferent and efferent. They are moving both sensory and motor information into and out of the spinal cord. All of these are afferent and efferent. We'll go over these at the end of today's lecture.
Sir,
Yep.
what about the
Nope. Just the roots and the peripheral nerves. That is it. It is quite complicated. You will see at the end of today's lecture what of those two I want you to know. We're going to focus today's lecture specifically on the peripheral nervous system. We're going to start with sensory and then move over to the autonomic nervous system. Starting with the card today. I already forgot. Cranial nerve number 7. On next week's lecture exam, next week, folks, I'm going to write them as Roman numerals. I mentioned that. I need you to be able to interpret Roman numerals. Which one is cranial nerve number 7? Facial. Is it sensory, motor, or both? It is both. When you understand that it's both, I need you to provide me a sensory function and a motor function. Let's start with the motor function. What are the motor functions of the facial nerve? What are you going to write for me? Facial expression, facial motor, facial muscles, something to that extent. That's what it controls. What about sensory? Taste, the sensation of taste. Done. You will tell me it's both and give me a functionality of both. I'm letting you know right now, since I'm not doing a review, we don't have time for a lecture review. One of these will be on your exam, lecture exam. That's going to tell you. All I have to do is change the number. It could be 5, 4, 3, 2, 1. Just going to change the number and ask you, is it sensory, motor, or both? What's its function? One of those will appear on your exam. What is the peripheral nervous system made of? So it's made of nerves. What else? Ganglia. Those are the two major organs of the peripheral nervous system. What are the two major organs of the central nervous system? Brain and spinal cord. These are the organs of those systems. They are, but they're not organs. Okay? They're part of the nerves. Just like here. We talked about what information moves through the dorsal root, correct? What is that blue neuron? It is sensory. What is the red neuron? Motor. You can see it recruiting the muscle. So the peripheral nervous system, yes, it has both motor and sensory. The only thing we're really doing on motor, because your lecture exam is going to have the muscles on it. So we already kind of covered small motor units, large motor units. That is part of the peripheral nervous system, are the neurons that recruit those muscles. We're going to focus on sensory today. And there are two groups of senses. Your general senses and your special senses. Your general senses, think of the senses you can feel at the tip of your fingers. What can you feel at the tip of your fingers, folks? What is it? Pressure. What's another one? Temperature. What's another one? Pain. What's another one? What in the world? Chemical. You can feel chemical with your fingers. You want proof? Shave a little bit of skin off. Touch some lemon. That detects acids. You ever notice when you touch soap, it feels weird? It's like actually you're using both touch and chemical. That slimy feeling is a chemical. You're recruiting a chemical receptor in addition to a touch. Don't forget touch, even though touch deals with pressure, vibration, pain, and stretch. Touch itself is another sensation. Special senses. What makes them special is that they have special organs to detect that sensation, like this one. What organ is that? The eye. What's your other special organ for senses? Nose. What's your other special organ for special senses? Ear. And the last one, not your mouth, tongue. What do your eyes detect? Light. What does your nose detect? Smell. What does your ear detect? Sound and balance. But what is it actually detecting? You're going to need to think about that. This one's a tough one, okay? Remember that when I ask you, what is it detecting after we discuss the ear? The tongue, what is it detecting? Taste, chemicals. Nose detects chemicals. Eyes detect light. Ears detect fluids moving. We'll talk about that. So here's the breakdown of the peripheral nervous system. The somatic motor. Oh, sorry. This is efferent. I forgot to write efferent. You have the somatic division. Look what it says. Skeletal muscles. The autonomic. This is what we're going to do the second part of lecture today. Division. This is smooth muscles, cardiac muscles, and glands. These are part of the peripheral nervous system. What we're focusing on today is the afferent division. And the afferent division is broken up into somatic and visceral. But what I want you to remember out of those is they both have general senses, touch, pain, stretch, pain, temperature, chemical changes, irritation, nausea, and hunger are general senses. The special senses have the organs. Hearing, equilibrium, vision, taste, and smell are the special senses. The receptors are the cells or the ends of neurons. They are cells or the ends of neurons that do the detection. Receptors detect stuff. They detect the environment, whether it's outside of the body or inside the body, the internal or external environment. Starters, you have your exteroceptors. These are your outer receptors. Most of these are going to be in the skin. These allow you to detect the outer environment so what's happening outside your skin when you touch stuff, but also what is happening to the skin itself. Is someone stretching your skin? They stretch it too much, it's going to hurt. They're detecting what's happening at the skin level. Exteroceptors. Interoceptors. These used to be called visceroceptors. They don't call them that anymore. These are your inner receptors, the receptors that are linked to all of these organs. So your inner organs, your inner environment. Does it hurt to breathe? My lungs hurt when I breathe. That's an interoceptor detecting pain. Can I feel my heart beating? That's an interoceptor detecting the movement. Oh, I think I'm gonna have a stomach bug. That's an interoceptor detecting some stomach irritation, intestinal irritation, discomfort. Interoceptor. Proprioceptor. This one's hard for students to comprehend. These are receptors that are typically going to be found in your tendons, in your ligaments, in the muscles, the skeletal muscles. What do these detect? Your body orientation. Is my bicep stretched? Is it contracting? What are my muscles doing? What position are they in? Your body orientation, relative position. Need to know that in order to know how to move. Okay, I still have a little bit of trauma. Give me a quick minute. I don't know why. I want to make sure that we are recording. Good. That last one was not fun. Microphone is on, don't worry. The receptors of the sensory system. These ones that we're talking about in the skin are all general senses, the type of sensory modalities you detect at the skin. The first type of receptor has this long name. It is a free nerve ending of a neuron. Sometimes they're just called free nerve endings. You will see both, okay? You will see both. So this neuron comes to the surface of the skin, it branches off, and at the very end of that neuron, there's the ends of the neurons. Each neuron is designed to detect something. Oh, detect a specific modality. What does that mean? Some free nerve endings, some neurons are going to detect what we call light touch at the surface of the skin. Some will detect temperature at the surface of the skin. Some will detect some level of stretch, pain. These can detect pain. Okay. And interestingly enough, they make what we call a receptive field. So that neuron is responsible for that field on the skin. You will learn more about receptive fields in physiology and how they can be used to help you locate where you're being touched, where you're being exposed to temperature, where your skin is being stretched, where there's pain. That is what free nerve endings detect. The rest of them are all named after old anatomy dudes, as I like to say. So I apologize because they're all named after somebody. The first one, Merkel's disc. Okay. So this one has this disc-like structure that sits underneath the epidermis of the skin. And a neuron is attached to it. This cell is specific to touch, what we call mechanoreception or receptor. It detects a level of touch. Merkel's discs are responsible for detecting light touch at the skin, including itch. Itch, believe it or not, is relative to pain. Okay, so these are specific to light touch and the level of itch. Receptor, yeah. It will be painful if it gets too itchy, but at that point you recruit other receptors. Yes, sir. It is the mechanoreceptor. So it's the disc that sits on top of the neuron. Okay. And it acts as a mechanoreceptor. They will be located right here, like at the very bottom of the epidermis. Those are all Merkel's disc. And they detect light touch at the surface and things that'll make you itch at the surface, like when somebody does that little feather thing. Brush it up barely against your skin and it itches. You're going to read they do more, but these are the two general senses that I want you to be familiar with for Merkel's disc. Meissner's corpuscle. This is another receptor. They're located right here. That's Meissner's corpuscle. This one is a gel surrounding the neuron on the inside, and the neuron is coiled. So what does this detect? It's the movement of the gel. These are designed to detect another form of light touch, or what we say light pressures on the skin, because it moves the gel when you get a little bit of pressure on the skin. It deforms the gel, therefore the neuron detects it. You can also say low vibrations at the skin. They're all a form of mechanoreception, some sort of touch. It is another mechanoreceptor. Mechanoreceptor, literally meaning it detects some sort of touch. There's one more touch receptor, another. It is called Pacinian. Pacinian's corpuscle. This one has layers of gel, and inside there's the neuron. So this one is found deep skin, almost in the hypodermis, still in the dermis. So what is this one responsible for detecting? Deep pressures, when someone's really applying pressure, force to your skin. High and strong vibrations. This one is located deeper in the skin. Named after scientist Pacinian. Pacinian corpuscle. Raffinis. These ones are interesting. Raffinis will have collagen or elastic fibers here. And the neurons, you can see, surround them. What those neurons are detecting is how much do those fibers stretch. This is a stretch receptor. How much is someone stretching your skin? That's what it detects. Stretch of the skin. Ruffini's corpuscle detects skin stretch. They also say skin sliding, like, you know, when you can feel somebody like, that's not really stretching, but when somebody moves, deforms your skin a little bit, they're stretching the skin, they're stretching the dermis. Ruffini's corpuscle. Not in the skin, still general senses. You have this neuron that'll wrap around muscle fibers. Or you have this neuron that wraps around tendons and ligaments. This one, if it's in the muscle, it is called a muscle, as simple as a muscle spindle neuron. It is a muscle that is around, a neuron that is around a muscle spindle. What does this one detect? When your muscle is stretched or when it is contracted, but it is detecting stretch. How stretched are your muscles? This is the one that elicits the reflex, folks. When you stretch your muscles too much, they want to contract. This neuron that wraps around the joints is called a joint kinesthetic. I already forgot what the other word is. It's not spindle. We're going to call it neuron for now. Joint kinesthetic neuron sounds right. Brain farting. What is this one detecting? How much your joints, your tendons, your ligaments are stretched. Both of these detect stretch. Just which part of the muscle is it detecting? Is it detecting the muscle at the joints? Or is it detecting the muscle at the muscle fibers itself? These are proprioceptors, folks. These give your body an idea of where those muscles are located, how they are stretched out. So when it comes to the exam next week, I'm going to ask you questions like this. So if I were to ask you, at the skin, which receptors can detect temperature? Free nerve ending is the primary temperature sensor at the skin. What if I were to ask you, which one does deep pressure at the skin? Pacinian. All right. What if I were to ask you which one detects if your skin is being stretched? Ruffini's. What are the general senses? Name one. Touch, pain, pressure, temperature, chemical. What are the special senses? Sight, smell, taste, hearing, equilibrium, balance. Let's go over the special senses. Yes. For question 3. I guess in that one, if it's a multiple choice, be able to determine, right, which of the 4 that I provide you are temperature receptors because I can provide you any of these 6, right? They're all external receptors. All right, we're going to go over the special senses. We're going to start with the olfactory system. Your sense of what? Smell. What is the special organ? The nose. What is scent? What are you detecting in the environment when you smell something like a rose? It is a chemical receptor. It is a chemical receptor. Chemical receptors can be general senses, like the ones at your skin, or special senses, like the one in the nose. Special organ here. So the products that you smell will release chemical particles. And those chemical particles flow into your nose and they get stuck in this mucus lining that is around your olfactory nerves. So there's this mucus coat that surrounds the free nerve endings there. They detect chemicals in the nose. The particles get stuck in the mucus. And guess what? Those free nerve endings will detect the chemicals. You can detect over 150 different chemicals. That doesn't mean you only detect 150 smells. When you start combining them, your brain interprets different smells. Okay. So they get stuck to the nose mucus. Here in pink are your actual cranial nerves. Those are the olfactory nerves. Then you have the olfactory bulb where they all connect to. That's how it's different. They all attach to the olfactory bulb. What cranial nerve is it? 1. Olfactory nerve. Okay. The special sense of smell is a chemoreceptor. Taste. Let's stop here. Tell me which ones detect taste. All right. Cranial nerve, which one detects taste? Cranial nerve 9. Which one's cranial nerve 9? Glossopharyngeal. Cranial nerve number 7 is which one? Facial. Both of those are responsible for your taste of food. Back when I learned this, they said, and there are 5 tastes. And they used to map them out in the tongue as follows. What are the 5 tastes? Sweet. Sweet used to say it was detected right here. What was the other one? Sour. What's another one? Salty. Another one? Bitter. Bitter and sour are, I don't know how they're different to be. I know how they're physiologically different, but I don't know how they taste. Like, I don't get it. Doesn't compute in my brain. What's the last one? Umami. What in the world does that mean? Savory. Think of like chicken or vegetable broth. Savory. Umami. Well, these are the 5 general tastes. And it turns out we really don't know how it's mapped out on the tongue. What we do know is that these 2 are interpreted by the facial nerve. These 3, the glossopharyngeal nerve. And this is for food. And I mentioned last week, there's technically one major other taste. Who knows what the other taste is? You need to be able to taste it. Let me see, what color haven't I used? We're gonna go with this bright green. Back over here. This is where your body detects taste. Water. And this one is controlled by your vagus nerve. It's not a true taste, but it detects water. You know, after you work out in the gym and you're like, I need water, and that sensation, it's the vagus nerve. Okay. Some people say there's one other taste that's missing. Something that we eat, that we taste. Almost everybody says spicy. What in the world is spicy? Does anybody know what spicy is? Spicy is mixing the salt, the sweet, the fat that comes in a chili pepper, but you also couple capsaicin. Anybody know what capsaicin does to your tongue? It makes it detect pain. So people are like, I can't handle spicy food. Guess what? That means you just have more capsaicin receptors than I do. People who can handle spicy foods don't have very many capsaicin receptors in their GI tract. Right? The more capsaicin receptors you have, the more you're gonna feel that spice burn. All it's designed for is to elicit a pain response. So the tongue uses a series of taste buds. These are all taste buds, and they are all projections in the tongue. They come in different shapes, different sizes, and they're all chemoreceptors. They detect chemicals. So what happens is when you eat your food, it has to fall in these little pits inside your taste buds. Poor hygiene, by the way, is going to make these smell horrific. Tongue hygiene is important. They will get stuck in there and rot away. At the very ends, you have receptors. Here's one of those receptors. At the very end, here are these receptor cells. And they have these free nerve endings. They look like little hairs just sticking out of them. When you eat a salt molecule, a sugar molecule, a sour molecule, a bitter molecule, an umami molecule, they're going to come in contact with the appropriate neuron, and it's a chemoreceptor. It's going to send that signal of taste to the brain. So it is a chemical receptor that binds to the receptors at the ends of your taste buds. combining certain amount of sweet, salty, umami, bitter, sour is what makes a good chef. They mix it all. They add things to help your palate, your taste interpret something that is flavorful. Chemoreceptors. This one's the beast. You ask me, this one's the beast. I think the eye is easier than this one. Your ear. Your ears are designed to detect what? Sound. What is sound? It is the waves of the sound in the air. So when I speak, what makes waves? What's making waves in the air? The movement of what? What's okay before it gets there? So between me and you, vibrations in what? The air. What's vibrating in the air, folks? What is it? Which molecules? All the air molecules in the air. Things we breathe. It is a fluid. Air is a fluid if it has molecules in it. No molecules, no sound. In a vacuum, no sound can be created. So it breaks my heart every single time I think about the Star Wars films when things blow up and make sound in the air. That will not happen. No sound in space, no air, no gases in space. So it is the molecules vibrating. When I speak, my vocal cords vibrate the air at a certain frequency. So the sound waves have a frequency, and that's going to determine whether it sounds low pitch or high pitch. Some people have high pitch voices, some people have low pitch voices. Depends on how my voice box vibrates that air to create the frequency that moves through the air. It goes into your ear, and it is first detected in your ear by vibrating the eardrum. We will not call this the eardrum. You will not call it the eardrum. This is the tympanic membrane. So that vibrates at the same frequency as the sound vibrates. Okay? So the frequency of the sound will determine how the tympanic membrane vibrates. Then that's going to vibrate these. These are the smallest bones in the body. There are 3 of them. These are the ossicles. As far as I'm concerned in lecture, we will learn them as the ossicles. Next week in lab, you will learn the actual names of each one. That's lab. Stapes or stapes. These will vibrate based on how the tympanic membrane vibrates. So you see how the sound makes the tympanic membrane vibrate. The ossicles vibrate. And what the ossicles are going to do is they're going to vibrate and send waves of fluid through this structure. This snail shell is called the cochlea. Hey, look at that. Vestibular cochlear nerve. The cochlea detects sound. So here's how it works. The ossicles will vibrate on this window, and they're going to move the fluids through the cochlea. The fluid is going to move through this chamber. We do not have to know the chambers for lab. I mean, for lecture, that'll be for lab. depending on the frequency, whatever that may be, the frequency will cut through these two chambers and leave the ear. It is lost. That sound wave will be lost. So you only hear it once. You only detect it once. When do you detect it? When it moves through the cochlear duct. Oh, and the cochlear duct is this one that I'm going to outline it in bright green. So you can see that it'll move through the cochlear duct. That's what the fluid waves will do in the cochlea based on the frequency of the sound. They will cut through the cochlear duct. And when it cuts through the cochlear duct, it'll move from here through this structure. This entire structure is called the organ of, I don't even know how to pronounce this. I just know this is some scientist. I don't know if it's Corti or Corti. I've heard both. Organ of Corti is the way that I'm going to say it. I don't know if that's true. Here's what happens. This one in pink does not move. It is stationary. This is called the tectoral plate, the tectoral membrane. It is stationary. When the sound moves through, it is going to make this membrane at the bottom. This one is called the basilar membrane, the basal membrane. Think of it as the bottom one. Basilar membrane. This one oscillates. Oscillates. What does that mean? When the sound wave moves through here, like this, tectoral membrane doesn't move. The basilar membrane will move back and forth as that sound fluid moves through the organ of Corti. As it moves through the organ of Corti, these cells in purple, they are called hair cells. Why? Because they have hairs that will attach to the tectoral membrane. Those in yellow are the hairs that are attached to the tectoral membrane. So when the basilar membrane oscillates, when it moves back and forth, these hairs bend. When the hairs bend, you detect sound at that frequency. So what you're really detecting when you detect sound is where are the hairs bending on the hair cells? Where are they bending in the cochlea of the organ of Corti? When they bend, it's going to send us action potential through the neuron. That one is your cochlear nerve. It will eventually become the vestibulocochlear nerve. So technically, do you see how the cochlea is coiled like a snail shell? If you were to stretch it out, it would be one long tube, just like shown below. So when the sound waves come in and they deflect the tympanic membrane, they make the tympanic membrane oscillate. That makes your ossicles oscillate or vibrate. they beat on the fluids in that window, and the fluids, based on the frequency, will move through the cochlea. So the frequency is going to determine how far down the cochlea it goes. If it has a high frequency, high pitched noises are going to cut through the cochlea duct early on. If it has a medium frequency, moderate frequency, it's going to cut through the cochlea duct like about halfway through. If it's a really low frequency sound, low frequency, oh, I forgot to write medium frequency. If it has a low like someone with a low voice, it'll cut through further along the cochlea. So the frequency of the sound of the fluids moving through the cochlea will be detected at different points. High frequency, medium frequency, low frequency. If the sound is a really low frequency, guess what happens? It goes this way. You don't detect it. It didn't go through the cochlear duct. If the frequency has too high a pitch, it'll just go out. It won't go through the cochlear duct. You can't hear it. This is why some animals can hear differently than we can. Their cochlea is a different shape, different size than ours. A dog whistle. Humans can't hear it. Too high a frequency. It does this. It just starts here, comes out, doesn't even get detected by the cochlea. But a dog surely hears it. Their cochlea is a different shape. It's wider here, right? And it is shorter over here, but it is like 3 times the length. So that's why they can hear better. Yep. It's all about how it's set up. Cochlea. Sound, folks. Sound. The vestibule. I say it includes these little loop-de-loops. They look like roller coaster loops. Vestibule. At the base of the vestibule, 1, 2, 3. You have 3 of these cupula. And the cupula is a gel-filled sac. So this is all like gel. It's a gelatinous structure that has hair cells in it. Here's 1 hair cell. Here's 2 hair cells, 3, 4, 5 shown. And their hairs stick up into the gel. The vestibule is designed to detect head acceleration. So changes in head movements. When does that happen? When you shake your head no. Your body's designed to detect that. When you're in a car and you stop and go, stop and go, stop and go, you're constantly accelerating, moving your head in space. When you move your head, the fluids in here, let me try that again. The fluids in here will slosh around in those. These are called, by the way, you're going to have to know this for lab. These are called semicircular canals, and there are 4 of them, 3 of them. There's fluid in there. So when you move, It's going to slosh around based on how you move, how fast you move, the acceleration. So when that fluid gets sloshed and it moves in a certain direction, because you moved your head in a certain direction really fast, it's going to make the gel bend. Therefore, it bends the hair cells. Can you see that bending hair cells is a big thing in the ear? Bending hair cells is what's going to allow you to detect sound. Bending hair cells is what's going to allow you to detect which way are you moving. So here's how a hair cell looks like. Here's the actual cell itself. And they have special cilia that want to stand up straight. And when the fluid moves, it's going to bend the hair cells in one direction, bending, or the other direction, bending. Your hairs will detect how much bending is there when you move your head around in the semicircular canals. So this detects head acceleration. That is what the vestibule detects. The vestibule is technically the cupula and the semicircular canals. So this detects head acceleration. How does this affect you? Well, you've seen people do those silly contests. I'm going to put a baseball down on the ground, put my forehead down, run around like crazy. This is what'll make you dizzy. Car sick, plane sickness, seasick. When you have too much information going to the vestibular system, when you're rocking up and down the boat, moving forward and backwards in a car when you're constantly stopping and going, accelerating, or on a plane. Okay. This is simply designed to detect how much you're moving and when. Think about it. It's only really meant to detect when you're running. But we've developed instruments to move us faster than running. That's why some people get dizzy and motion sickness. The otolith are part of, or the saccules, this region right here. This region, some folks still call part of the vestibule, and that'll work. That's all right. I'm not going to judge. They know better than I do. But they work a little bit differently than the vestibule. The saccules detect gravity. Your otolith is a structure, that purple structure in there. And inside that purple structure, you once again have hair cells, hair cells, hair cells. What do the hairs do? They stand straight up into this gel. That's a different gel. It is different than the vestibule, only in this fact that gel is full of calcium, little calcium rocks, crystals, I should say calcium crystals. So this is what they look like. They stand up straight when you're standing up straight. When you bend over to tie your shoes, what happens? That gel kind of bends down in the direction of gravity. Why? Those calcium crystals have a weight to them, so they drag the gel down. what happens to your hair cells? They get bent. They bend in the direction of gravity. So even your otoliths have hair cells. They detect gravity. Are you laying down face forward? Are you laying down with your back up? With your back on the bed? Are you in a weird position where you have your feet hanging on the ceiling and now your head's upside down? They will detect the orientation of your head by detecting gravity. This is what your otoliths do. All three of these in the vestibule. In the cochlea. Vestibular cochlear nerve. Remember that. Hearing and balance. What do hair cells detect? This is a trick question, believe it or not. How do they detect the movement of fluid? They detect how much the hairs are bent. This is called deflection. They measure deflection, which, yes, it will be moved based on the fluids that surround it, whether it's a gel, whether it's in the cochlea, right? They detect deflection. And this deflection allows us to use which sensory systems? Hearing, balance, or equilibrium. The vestibular cochlear system. Why can we detect low versus high frequencies? Well, it depends when the sound fluids, I should say when sound fluids move through different regions of the cochlear duct. This is a hard concept to understand. And what I want you to understand is as the sound moves through the cochlea, I'm going to go back a couple of slides. It cuts through the cochlear duct. Here it is, right here. As the sound moves in this direction, it will cut through the cochlear duct at some point. This region in gray, you can see, is the cochlear duct. The shape of the cochlea is different as it moves through the cochlear duct, which tells certain frequencies to cut through at specific points, allowing your ears to detect different frequencies of sound. One of the things that I forgot to mention that I think is important to mention, if you hear sounds too much, like if you work around heavy machinery, if you go to a lot of concerts, do you know what happens to your hair cells? They will break the hairs. The hairs will break after being exposed to high amplitudes, loud noises. After a concert, if you're really close to a speaker, you're going to hear ringing for a day or two, if you're lucky. If you're not lucky, and if that hair cell is permanently damaged, You're going to hear a ringing for the rest of your life. What is that called? Tinnitus. What's that? What do you mean? What do you mean by inflammation? I don't know what you're talking about. Tinnitus, tinnitus is an inflammation, but tinnitus in this case is the damage to the hair cells. So there's nothing allowing that to connect. It's going to be filled with fluid. Okay. No, but that is the main reason that'll cause ringing in people that are working around heavy machinery, is damaged hair cells. They just deteriorate over time. Function of aging. We all have to look forward to. The eyeball is the major organ that allows vision. There's a few terms that I'm going to ask you, very few terms compared to lecture. We're going to start with the window of the eye. This is where the light will come through. When light goes through the eye, it's going to start by going through this structure that I've outlined in blue. That is the window, that is the cornea. You have a special set of muscles right here. This muscle will open and close this space. The space here is called the pupil. That is the space the light runs through. The muscles are called the iris. They will open and close the pupil to allow more or light in. The iris gets to choose how much light goes in, how much light goes through, a little bit of light or a lot of light. The iris is also going to be the structure that gives your eye its color. So if you have blue eyes on the surface of the muscles, it's blue. Brown eyes on the surface of the muscle, there's brown pigment. Not like that, because what the pigment has to do is block the light. Okay. So when the light comes through, only the light that goes through the pupil will be able to come through. The iris blocks the rest of the light. So the light comes in as a beam. And then it hits the lens. The lens's job is to bend the light and focus it on the back of the iris. Not the iris, the retina. And it's going to focus it on this area right here. And this is called the fovea centralis. This is where you want the light to focus. What happens if the light doesn't focus at that point? You get blurry vision. That's when you need glasses, because the light isn't bending right through either the cornea or the lens, or both. So the cornea helps the light move through into the lens, and then the lens does something very special. The lens does something that I say, it bends and refracts the light. So it can focus on the fovea centralis. The fovea centralis is on this layer of the back of the eye. Everything in that orange pink stuff is the retina. The retina holds all of your light receptors. What's interesting, this is what the lens does. If you see an A like this, in that orientation, the lens is going to bend it, the lens is going to refract it. So when the light gets over here, the A now looks like this. It's upside down. So your brain sees the world upside down in a mirror image. The brain has to rearrange everything instantly so you can see the world in the correct orientation. That's what your occipital lobe does. Your occipital lobe rearranges, reorients you to the world as it is. Some people have challenges with that. And things like words and letters, numbers, don't get rearranged appropriately. What is that called? Yeah. It's some challenge with the light getting in and the light being interpreted by the occipital lobe that gives some people the challenge of dyslexia. Okay. So when it goes in here, it'll eventually end up in the occipital lobe. That is where your visual cortex is. Keep in mind, it can be other things too. But the occipital lobe is one of the main causes. Okay. That is the pathway the light gets to get to the photoreceptors on the retina. The fovea centralis is the concentrated area of photoreceptors. That is where you want the focal point to be. Okay. So the light has to go through this structure. What is the outer wall? What is the outer window called? This region that I'm outlining in pink, this is the iris. Look at that person has brown eyes. But that is a muscle. It is going to open and close how big the pupil is. The pupil is the space that the light has to get through. Darker pigmented colors are a little bit better at blocking the light. If you have light colored eyes, like light blues, the light greens, people like that are more prone to get light damage to their eyes. You have light blue eyes, light green eyes, sunglasses, and the expensive ones for your eye care. The lens, what it does is it allows you to focus. Your body's going to physically change the shape of the lens. That is how you can focus on things that are far or focus things that are close. Your body has to bend those lenses, okay, to allow you to focus the light on the fovea centralis. Remember, this fovea centralis is part of the retina. That is where your photoreceptors are at. One fun fact, and you guys are going to be like, this is gross next week, and I think it's the coolest thing. This is filled with like a clear jello. When you cut the eyes open, it's going to be full of gel. It's called the vitreous humor, not important for lecture. But if you see those little floating things every now and then, you're like, what the heck are those floating squiggly lines? It's debris that's just floating in there. Impurities that are floating in your gel. You'll see them. Some people have more than others. Remember that gel when you open up that eye. Humor. When the light hits the retina is a structure that still like blows my mind. It is a layer on the back of the eye. That is the retina. The light comes in from this direction. And interestingly enough, as the light moves through, it has to move through the blood vessels. It has to get through the alpha, beta, polarized cells. Light has to travel through that before it finally gets to the photoreceptors. The photoreceptors are back here. I remember when I read about this, I was like, how the heck does the light make it through everything and still get to the photoreceptors? Photoreceptors are pretty gosh darn sensitive. They're really good at doing their job. You have two flavors of photoreceptors based on the shape of the opsins. It's this region of these receptors. You have ones that look like this. These are called rods. This group of photoreceptors, they're not rods, they're cones. Your eye, your photoreceptors will be made of both rods and cones. And they detect different types of light. Please, for the love of God, tell me these detect light. Especially if it's a free response. Very likely to be. So when they detect the light, the rods detect light in a black and white. They detect black and white light. How much light is there? Is there a little bit of light, a lot of light in that general region? That allows you, rods, to detect visual resolution. That's how you can see shapes, crisp, clear lines by the rods. People don't understand that. Have you ever seen a black and white movie? You can still see where someone's coat, where someone's hat, where someone's eyes, the areas where they start and end. Just with black and white. That is what rods do. They allow you to determine the spatial resolution. Cones do something different. Cones detect color, light. I forgot to put light. Look at me, I already failed. Resolution. Both of them detect light. Cones detect a wavelength of light. Lights are photons that are moving through the environment at a particular wavelength. They detect certain wavelengths of light. And they are optimized to detect three major colors. What are the three major colors your cones? Sorry, let me say that again. What are the three major colored light waves that your eyes, and I mean your cones in your eyes are detecting? What are the three major colors, folks? RGB. Red, green, blue. There's a reason why TVs are built to show these colors. Many people think it's red, blue, yellow. Primary colors. Not what we see. Okay. So how does that work? Well, your blue cones are designed to detect the wavelengths at 420 nanometers. That allows you to optimally see blue light. But you detect this whole range with your blue cones. Doesn't mean you detect them all well. You detect blue the best with blue cones. Green cones detect this range from here to here. Oh, wrong one. To here. Green cones. That doesn't mean they detect them all well. They detect green the best. Red cones detect from here to here on the color spectrum, but they detect red the best. Using a combination of these three cones, we can see the full spectrum of the rainbow. Rods, remember, they're detecting black and white light. These are the colors from this region to this region that help you interpret resolution the best. Okay. Almost everyone is born with rods. Rods are very important for resolution, but there are some people who are born without certain cones. They just don't have them. It's not in their genetic makeup. What do you call that? So to a person who's missing the green cones, green is best detected by the green cones. Even though the blue cones and the red cones overlap in the green region, that person is going to detect everything in green and some yellows as gray because they do not have the green cones to best detect green light. So they're going to see the world very differently than someone who has all three types of cones. You will see everything in red as gray. Or parts like red, orange for sure. You're going to see it as gray. Blue and purple would be gray. Correct. Not the rest, not this. Yeah, it's the primary ones that you will see in gray. Okay. Out of funsies, we see in three major colors, right? Our cones come in three major colors. That's the better way to say it. That allows us to see the rainbow. Some animals see black and white. What animal sees in black and white? How many colors do they see? They see two colors, but they can't interpret colors. They have a blue and a yellow cones, but to them, the world still looks black and white. Not well. Not well. Does anybody know which animal has like the most craziest cones? They have 9 cones. They can see ultraviolet light and infrared light. It's like the craziest thing ever. Mantis shrimp. Good job. If you look at their eyes are the craziest looking things. And nobody can we as humans cannot comprehend how they see the world. There's no way to do that. Their brains are just crazy. Forward. How are you going to answer this on next week's lecture exam? So these detect what type of light wavelengths? They are designated to optimally detect red, blue, and green. Remember, that doesn't mean they only detect red, blue, and green, or else we would only see red, blue, and green. They're designed to optimally detect those colors. They detect other colors in between them. What does a rod detect? Light wavelengths. In which color? This one is black and white. And this one, again, is important that you understand. It's going to allow you to see resolution. People can be born without these. They won't see colors, but they can still navigate the world with vision. It'll just be in black and white. The nerve plexuses. So here again, the main thing I want you to be familiar with are the roots that come off of the spinal cord. That is where the peripheral nervous system starts. And the peripheral nerves. There are, I think, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 peripheral nerves I need you to be familiar with. Starting with what comes off the cervical plexus. The cervical plexus is made of the spinal nerves C1 through C5. The brachial plexus, that one goes to the arm. It is made of C5 to T1 spinal nerves. They are a group of nerves, a plexus, that go to the arm, and they make the individual arm nerves. You have the lumbar plexus. This one primarily goes to the thigh. The lumbar plexus is made of L1 to L4. The sacral plexus overlaps. It starts at L4 and goes down to S4. From the cervical plexus, I need you to know one. I apologize. I don't know what happened to these symbols. Okay. I need you to know the phrenic nerve comes off of C3, C4, and C5. Those make the phrenic nerve. The phrenic nerve is responsible for powering your diaphragm. Guess what happens if you damage C4? It was nice knowing you. You do not want spinal nerve damage at that range, or else you're going to have to put on some iron lung device. Yep. The phrenic nerve is responsible for the diaphragm. And as silly as it is, I say C3, C4, C5 to stay alive. That's the only one I got. The rest of them are tough. We have to remember it. Cervical plexus, that's the only one off the cervical plexus, is the phrenic nerve to the diaphragm muscle. The brachial plexus has 4 nerves. We're going to start with the musculocutaneous nerve. The musculocutaneous nerve branches off of C5, C6, C7. Those are part of the brachial plexus. They will come together and make this nerve and the musculocutaneous that is responsible for the flexor muscles in your arm. What is one flexor muscle in your arm, folks? Your biceps brachii. That one is controlled by the musculocutaneous nerve. Which one are we doing next? The ulnar nerve. This one is C8 and T1. That's where its roots are. And it goes down to the muscles that flex your wrist and fingers. What is wrist? What is finger flexion look like? Closing the fist. That is powered by your ulnar nerve. Extension of the fingers is a different nerve. Opening the wrist is a different nerve. Fun fact, the ulnar nerve is your true funny bone. The funny bone is not a bone. When you whack the back of your elbow, you are pinching the ulnar nerve about right there. And it sends a really weird sensation up and down your arm. Sometimes you feel it in your shoulder, sometimes you feel it in your neck because of how it moves through the body. Because you're technically pinching the nerve at the bone. So people just call it a funny bone. The radial nerve, this one is a very complex nerve. So complex, it has a bunch of roots. It starts at C5, C6, C7, C8, and T1. Pretty much the entire radial brachial plexus makes the radial nerve. This one is responsible for your extensor muscles in your arm. Triceps brachii. And the extensor, it extends the wrist and fingers, opening the fist. That is the radial nerve. The axillary nerve is C5 and C6. Those are the roots of the axillary nerve. It powers your shoulder muscle. What's your shoulder muscle called? Your deltoid. It is powered by the axillary nerve. All of those four, these two, brachial plexus, these two arise from the brachial plexus going to the arm, forearm, and fingers. The axillary nerve stops at the shoulder, the arm. Lumbar plexus. I think I'm only making you do two. Sweet. Only two. Interestingly enough, they both have the same roots. L2, L3, L4, L2, L3, L4. But they come together and they become different nerves that power completely different areas. The femoral nerve powers the flexor muscles in your thighs. Oh, not thigh, thighs. Does anybody know what that muscle group is called in the front of your thighs? Your quads. Your quadriceps. The obturator nerve also starts at L2, L3, L4, and it powers the medial thigh. This one makes you adduct the thigh. Can you think of a muscle that adducts the thigh? It's on your muscle list for lab. Gracilis. Excellent. And adductor magnus and longus. Those are on your list for lab. The obturator nerve powers those muscles. Again, I apologize about the symbols. I don't know what happened. And it only happened at this part. This one's a fun fact. The tibial nerve. Let's start with the tibial. I'm going to label these ones in red. L4, L5, S1, S2 to S3. Did I put flexor up here? Oh, I see what I did. Got it. Got it. This one is responsible for the flexor muscles on the posterior compartment of the thigh. Your hamstrings. What muscles make up your hamstrings, folks? Who can name one of them? Back of the thigh. Biceps femoris. Semitendinosus. Semimembranosus are powered by the tibial nerve. And then it goes to the posterior leg. What muscles on the posterior leg? The soleus, the gastrocnemius. It is controlled by the tibial nerve. The common fibular starts at L4, L5, S1, S2. It also does the hamstrings. They work together to do the hamstrings. And then at the leg, it does the anterior muscles on the leg. Who can name one muscle on the anterior leg? Tibialis. Tibialis anterior. This is where it's weird. This region right here, the tibial and fibular nerve, anatomists used to think that was a whole separate nerve. That is the sciatic nerve. The sciatic nerve is not an individual nerve. The sciatic nerve is made of the tibial and common fibular nerve here at the thigh. Why? Because they work together. And then they split up when they get at the knee. As simple as this. Muscle compartment of the phrenic nerve, diaphragm. Which plexus? Cervical. Which roots? C3, C4, C5. I want you to be able to tell me which nerve innervates the biceps brachii. Which nerve innervates the biceps brachii, folks? The anterior arm. Musculocutaneous. Okay, there's 10 of them. We can do it. We can do it. This is a typo that I gotta fix. This was supposed to be T1. T1. I've been known to ask very similar question 1. Question 9, sorry. If these are your vertebrae at T1 and T2, you have a spinal nerve, a root that comes out on the left and right side. Some patients, their vertebrae starts to grow extra bone and it'll pinch that spinal nerve. That is horrific because it can be excruciatingly painful. And you will have to know if this is T1 spinal nerve, which plexus is it going to affect? The brachial plexus. Your patient may feel pain in their arm, even though there's nothing wrong with their arm when this happens. When it's pinching a spinal nerve. This could happen after an accident. After a really bad accident, God forbid, your patient has severe damage to T8. What are their consequences going to be? Which plexus is no longer going to function? Which plexus, folks? All of the nerves that come off the lumbar plexus. The femoral obturator. There's more. All the nerves that come off the sacral plexus. Sciatic, tibial, fibular, sural. All of them. Based on where the damage is. These would not function. They're going to be on a wheelchair. The peripheral nervous system. Our autonomic nervous system. Before I move on, is this afferent or efferent? This is efferent. That means your autonomic nervous system is responsible for motor responses. To be specific, visceral motor. That means things you do not control that are automatically controlled. One branch is the sympathetic division. Look at this dude. What parts of the body are controlled by the sympathetic division? When you experience a fight or flight response, what has to happen in order for you to elicit a fight or flight response? Okay, so you're in danger. What do you do when you're in danger? You should be doing some sort of exercise, running away from a predator, fighting a competitor, or hunting down prey. This doesn't happen in the human world, right? Stress. Next week we have an exam. Fight or flight response. You're welcome. What has to happen to support a fight or flight response? What is that person's body going to do? What about heart rate? Your heart rate goes up. What else goes up? Your breathing rate. What else goes up? Your blood pressure. What else has to happen for you to support that type of activity? When you exercise, think about jumping on the treadmill. What happens to you shortly after you jump on the treadmill? What else do you increase? You need to cool off. These are all governed by the sympathetic division of fight or flight response. Parasympathetic. We should all be doing this right now, but we signed up for this. This is the rest and digest response. Rest and digest. What is that person's body going to do to elicit a rest and digest response? Decrease heart rate, decrease blood pressure, decrease breathing rate. What is it going to do to sweat production? Nothing. You don't need it. Your sweat's naturally not producing anything. So it does nothing to sweat. What does it increase? Digestion activity. Enzymes. acid production. Peristaltic contractions, moving the food through. That is a rest and digest response. The sympathetic division. This means short. What does that mean? Well, here you have a neuron that is recruited in the spinal cord. The preganglionic, those neurons in red, are short. Sympathetic means the preganglionic, the first neuron that leaves the spinal cord. The preganglionic neuron is short. So here's the neuron. It leaves the spinal cord. It is short. Then it's going to go into the ganglion. What is the ganglion? The ganglion are these clusters. So since that neuron occurs before that cluster, that is why it's a preganglionic neuron. Then you're going to recruit the next one, the one that I'm drawing in red. So here's the next neuron in red. That is the postganglionic neuron. Guess what? This one is long. That is what sympathetic means. Short preganglionic neuron, long postganglionic neuron. This is unfortunately why it sounds very similar to parasympathetic or sympathetic. Sympathetic is describing how this is arranged, how it's set up. Let's do parasympathetic. Parasympathetic has a long start. What does that mean? Let's go over here. The neuron is recruited at the medulla, the pons, or at the end of the spinal cord. Look at how long the preganglionic neuron is. Sometimes it goes all the way to the organ. Sometimes it almost gets to the organ before it gets to the ganglion. There's the ganglion, there's a ganglion there, there's a ganglion there, there's a ganglion there. So the preganglionic neuron is long. Preganglionic neuron is long. Here it is coming out of the spinal cord or the brain, and it is super long. It gets to the ganglion. The ganglion are in blue. And then it has a itty bitty postganglionic neuron. Some of them are really short, some of them are a little longer, but it has a short postganglionic neuron. So they're arranged differently. They do different things. Parasympathetic, rest and digest. We should all be doing that right now. Next week, you guys are going to be like that dude. Hopefully not. Fight or flight. Both will affect a target organ, like the heart, shown below. The parasympathetic, please remember, rest and digest. Sympathetic, fight or flight. How do they do this? Here is the post ganglionic neurons for both of them. The parasympathetic response is shown in a neuron in blue. That neuron is going to send an action potential down its body, and it's going to release a neurotransmitter, the purple one, on the heart. The neurotransmitter is technically sometimes called a drug. The neurotransmitter released by the parasympathetic nervous system. We should be familiar with it right now. It's abbreviated ACH. Which one was that one? It is acetylcholine. If you put acetylcholine on the heart, it is gonna stimulate a rest and digest response. Acetylcholine tells the heart rate to decrease because of the receptors that are on the heart. The sympathetic system is going to release a different neurotransmitter. Does anybody know what neurotransmitter is released by the sympathetic division? It used to be called adrenaline. It is now epinephrine, an EpiPen. You can abbreviate this one as epinephrine or norepinephrine. They are both neurotransmitters. One is a neurohormone of the sympathetic division. So this postganglionic neuron of the sympathetic division is going to send an action potential down. And here it's going to release the epinephrine. The epinephrine are going to elicit a flight or fight response. Therefore, they're going to increase heart rate. Both will tell the heart what to do. One will increase heart rate, one will decrease heart rate. One uses acetylcholine, one uses epinephrine. What is it going to do? What is acetylcholine going to do to the intestines, folks? Increase activity. Increase the enzymes, increase the acid production, increase the movement of those muscles. That embarrassing movement that happens after you eat, you hear your stomach, it's like starts talking to you. What is epinephrine gonna do to your stomach? Decrease activity. Decrease activity of the intestines. Your fight or flight response. This one can literally scare the shit out of you. When are you going to decrease your breathing rate in a fight or flight or a rest and digest response? Which division is responsible for rest and digest? The parasympathetic division. When you sweat, are you in a fight or flight or rest and digest response? Fight or flight. You need those muscles. You need that sweat to support your muscle activity when you're running away or fighting. Is this a sympathetic or parasympathetic response? Sympathetic. What neurotransmitter is used by the parasympathetic division? Acetylcholine. The sympathetic division? Epinephrine. What response does acetylcholine promote? What are you going to tell me? rest and digest. The sympathetic pathway, if we recall, starts in the lateral horn. And I mentioned that the lateral horn in the spinal cord is responsible for visceral motor responses, things that you do not, things that you are automatically controlled in your body, involuntary muscles. And look at the pathway they take. They start here, and that neuron goes out of the lateral horn into the ventral root. But then it does something really bizarre. When it enters the spinal nerve, it goes into this ganglia. This ganglia is the sympathetic, only in the sympathetic division. It is the trunk ganglion. And in the sympathetic, this preganglionic neuron is short. So it ends right there. That is where most of the sympathetic neurons end, the preganglionic end there. Short preganglionic neuron. So the second one, post, starts here. So it recruits another neuron. And look at what this neuron does. It comes out this way, and it goes back down the spinal cord, and it'll either go out to the front, to the back of the body, or the front of the body, depending on where it's going to. So it has to take this loop-de-loop around the sympathetic trunk ganglion. Only in the sympathetic pathway. What is really bizarre is what it can do to get to the eyes. Here it starts at the lateral horn. That neuron goes down the ventral root into the spinal nerve, into the sympathetic trunk ganglion. And look, it can go up. For example, to get to your eyes. That's what it does. The sympathetic trunk ganglion runs along the spinal cord, so the neuron can start here and then go back up to get recruited at the sympathetic trunk ganglion to get to your eyes. What is it going to do in your eyes? It is going to affect the iris. How open are the pupils? It is going to affect your tear ducts, your salivary glands, your sweat glands. Think about what it wants to do in a fight or flight response, because that is the sympathetic pathway. It's going to affect your heart. It's going to affect the lungs. It can affect many organs in the body. Let's talk about some of them. The iris. We're going to repeat this a lot because I want you to get this right. The sympathetic system division, is it rest and digest or fight or flight? What is the neurotransmitter? When you put epinephrine onto the pupil, look at what the pupil did. Dilated. What does that mean? It opened up. In the fight or flight response, when you're running from a predator, having to fight a prey, do you want more light or less light going into your eye? Why do you want more light going into your eye when you're fighting a predator, fighting a competitor? So you can get more information, more visual information. You're going to be a better fighter. So what does it do? You tell me. Dilates pupil. The iris opens up. Parasympathetic, rest and digest or fight or flight, folks. Which is the neurotransmitter? When you put acetylcholine onto the iris, it constricts the iris. It doesn't completely closes, but it, what's the better word to use? Constricts. The better word is shunts. Makes it smaller. Think about it. When you're going to sleep, do you want a lot of light going into your eye, even though your eyelids are closed? No. You want less light going into your eyes as possible. Therefore, you're going to tell me it constricts the pupils. It makes them smaller so you can go into a rest and digest response. The sweat glands. These are the ones to cool you off, okay? Specifically the ones to cool you off. The sympathetic division, fight or flight, rest and digest. Fight or flight. What is the neurotransmitter? So when you put epinephrine on the sweat gland, what is it going to tell the sweat gland to do? Release the kraken. Produce sweat. That's going to support your exercise. Parasympathetic. Which response? Neurotransmitter. When you put acetylcholine, what is that going to tell the sweat gland to do? The sweat gland's normal state is not releasing sweat. So it tells the sweat gland to do absolutely nothing. Stay that way. One of the most complex one right here. Whew. Here we go. The parasympathetic system. Let's start with the parasympathetic. No, let's start with the sympathetic. Nope. Let's start with what the urinary bladder does. What does the urinary bladder do? Holds the urine. So when this is filled with urine, okay, you do not want to trickle pee everywhere. That would be embarrassing as all hell. So your body has this muscle right here. This one is the internal urinary sphincter. While this fills up, this one remains closed. So urine doesn't exit into the urethra. Makes sense. You don't want to, again, you don't want to be trickling pee as the urinary bladder is filling up. That would be embarrassing. In nature, that would attract the predators right to you. You're leaving a trail right to you all the time. So what the sympathetic nervous system does is it releases a fight or flight response using epinephrine. But epinephrine, the neuron only affects this sphincter. That is the interesting thing. The neuron of the sympathetic system only goes to the internal urinary sphincter. When you put epinephrine on it, do you know what it does to that? Counterintuitive. It makes that one relax. Weird. When does it do it? When it's time to pee. Only then. It won't put epinephrine on it any other time. Only when it's time to pee. The parasympathetic, fight or flight, rest and digest. Another counterintuitive. Well, this one is acetylcholine. Acetylcholine is going to affect the muscles on the urinary wall, urinary bladder wall. When is it going to release acetylcholine? When it's time to pee. What is it going to tell these muscles right here to do? Contract. When you contract, you're going to force that urine out. So it makes the urinary bladder muscles contract. This is a weird setup. Your body is going to use both when it's time to pee. Whether you like it or not, you do not have control over these. These are going to be told it's time to pee when this fills up. When it fills up, it stretches those muscles. It's time to pee. It's time to get rid of the urine buildup. Those both are involuntary, controlled by the autonomic nervous system. Thank God we have one more set of muscles down here. These are your external urinary muscles. These are skeletal muscles. What does that mean? Those are the ones where you're like, oh God, 5 more minutes before I can go to the bathroom. These are voluntary. Those are the ones you have to train to hold the pee, so you don't have an accident. Takes kids a while to learn how to figure that one out. You need to learn how to control that one. Okay, so this one is somatic motor, not autonomic. The other two are controlled by the parasympathetic and sympathetic division when it's time to pee. What does the sympathetic fight or flight response? Which neurotransmitter, folks? What is, because I can ask you that, what does epinephrine do to the urinary bladder? It's going to relax what? Internal urinary sphincter. Not intuitive. Relax, but it's fight or flight. It's all about the receptors, folks. All about the receptors on those muscles. What does the sympathetic system stimulate on the glands associated with sweat production? What does acetylcholine do to sweat production? Nothing. Again, this is the eccrine sweat glands. Apocrine sweat glands, a different rule. I don't need you to know it. Some of these are counterintuitive. I remember in physio, we get into more details in the autonomic nervous system. I remember this. One student asked me, wait a minute, how is this system involved in sexual arousal? They're like, rest and digest. When I'm aroused, I'm all hot and heavy. I tried my best not to laugh. It didn't work very well. Because the way he said it, trust me, if you were there, he was legitimately confused, but trying to make a joke at the same time. So it got the best of me. Well, it makes the blood vessels relax. Why is that important? Why do the blood vessels need to relax during arousal? Blood flow to erect the penis. That just helped those muscles relax so erection can take place for sexual arousal to occur. Sometimes these things are counterintuitive. For the sake of lecture, these are the parts that I want you to be able to tell me what happens. Parasympathetic response, acetylcholine. Sympathetic, epinephrine. What is acetylcholine going to do to the heart? What is it going to do? Acetylcholine. I'm not going to necessarily ask you parasympathetic or sympathetic. I can say acetylcholine or epinephrine. What is epinephrine going to do to the heart? We did this one as a group. We did this one as a group. This one should make sense for the lungs. Digestive tract, we talked about it. Blood vessels, I need you to think about it. What is it going to do? Depends is the right answer, right? What is it going to do to the adrenal gland? By the way, this is why it used to be called adrenaline, because the adrenal gland releases a lot of epinephrine. What is it going to do to your fat tissue? What will? Yeah, when you're in a fight or flight response, you want to turn that fat into sugar. The one I forgot to include or got deleted was the urinary bladder. Don't forget the urinary bladder, okay? But we talked about that as a group. All right, next week, you know what's coming. Don't forget your Scantron. Okay, don't forget your Scantron. By the way,